Hello my dear aspirants, I hope you all are doing well and I hope your preparations are also going well. Today I am again back with a very important topic for your CSIR NET or any kind of life science exam. So watch the video till end, and if you don't understand any specific point in the video, then you can comment down below. If you want to take detailed coaching from me for CSIR NET, Northeast LATE or any life science exam, then you can contact in the given number or you can comment down below. So without wasting much time, let's start the video. So ELISA, which is also known as enzyme-linked immunosorbent assay, it is a technique which is used to detect antibodies, hormones, peptides, proteins, antigens in the blood. Okay, so there are different types of ELISA which you need to study, that is direct ELISA, indirect ELISA, sandwich and competitive ELISA. Out of these four, competitive ELISA is a little bit different, that also we'll discuss. So direct ELISA is done to detect antigen. Indirect ELISA is done to detect antibody. Sandwich ELISA is done to detect antigen. And competitive ELISA is done to detect antigen or antibody, but it is done in a case where antigen or antibody concentration is very low. In case of very low concentration, competitive ELISA is performed. In order to perform ELISA, a specific plate is used which is made up of polystyrene, and there will be like 108, 96 or 56 wells, and that specific microtetra plate will be used in order to perform any of these ELISA. So direct ELISA, as already mentioned before, is performed in order to detect any presence of any antigen in the blood sample. Suppose a patient is having a particular antigen in his blood sample, then what will happen? See, the blood sample, the antigen from the blood sample will be captured in that well. Okay, when the antigen will be captured in that well, and then after each step washing will be performed, and if he is having antigen in his blood sample, then even after washing, this antigen will not get washed off. Okay, then what will add? Will add antibody which is conjugated with a specific enzyme. So this antibody is called enzyme conjugated antibody. So this antibody, which will be specific to that antigen, will be poured in that well. Okay, if there is a presence of antigen, then only the antibody will bind with the antigen. If the patient is not having that antigen in his blood sample, then the antibody will not get bound to anything. Here the patient is having the antigen, that's why the antibody got bound to the antigen. And what is the role of this enzyme? This enzyme is the main thing when we are studying ELISA, because here the E stands for the enzyme only, this enzyme. Okay, so why we have conjugated the antibody with enzyme? In order to detect something. What will detect? In the second step, we will add one substrate. You all know that all the enzymes have their specific substrates with which they have the affinity to bind. So after that, we'll add one chromogenic substrate. Chromogenic substrate means any substrate that will produce a color. When it will produce a color? When it will bind with its specific enzyme. So we will add one substrate which was specific to this enzyme. If there is the presence of enzyme, then only this substrate will show us color. Now, because this patient was having the antigen in his blood, that's why the antibody specific to the antigen got bound with the enzyme. And because the antibody is there, even after washing, again after this step also we'll perform washing. If the antibody is bound with the antigen, then it will not get washed off, because there will be affinity, there will be a bond between these two. Then only the chromogenic substrate will bind with the enzyme and it will give us a color. And at last, if we see color, then we will come to know that antigen was there, that specific antigen was there in the patient's blood. What if the antigen was not there? If there is no antigen, then after washing, there will be nothing. After washing, the antibody will also get washed off along with the enzyme. Then even if we add chromogenic substrate, no color will be seen, because the substrate will show us color only if it is bound with the enzyme. I hope direct ELISA is clear. Now coming to indirect ELISA. Indirect ELISA is performed in order to detect antibody. Okay, so if we have a specific antigen in our blood, then only we will have the specific antibody in our blood. Okay, So ultimately here we, in all cases, we are identifying the specific infection in the blood, in the body. Now in order to perform indirect ELISA, first we will take those wells, that means antigen coated wells. Here previously, here the antigen is not coming from the patient sample. Here already the antigen was coated in the wells. Then what we will add? Here we will add the patient sample. If the patient sample is having that antibody, then only it will bind with the antigen which was already present in the well. Okay, ready-made wells here we are using where already antigens were coated. Then we have added the antibody from where? From the patient sample. So if the patient sample will have antibody, then it will bind with the antigen. Then again, every time we are performing washing steps, because the patient sample was having that antibody, that's why it was bound with the antigen. Then what we will add again? We will add secondary antibody which will be conjugated with enzyme. So here we were using only one set of antibody conjugated with enzyme, but here we are using another antibody. This secondary antibody we are using artificially. This antibody is not from the patient sample. The first, that is the primary antibody, was from the patient sample. Secondary antibody, why we are using? Because this secondary antibody is specific to the primary antibody. If there is presence of primary antibody, then only secondary antibody will go and bind with the primary antibody. If there will be no primary antibodies in the sample, then the secondary antibody will get washed off after the washing steps. But here there is presence, so both of them are bound together. Here enzyme will be conjugated, then we'll add chromogenic substrate specific to the enzyme. If there is presence of everything, then only chromogenic substrate will show us color, and by detecting that color we will be able to know that the primary antibody was present in that sample or not. If it is present, then only secondary antibody will bind, and then only the substrate will bind to the enzyme, and then only color will be there. If there is no color, that means there will be no primary antibody in the sample. Okay. Next one is your sandwich ELISA. Sandwich ELISA is also used to detect antigens. Here, like in the indirect ELISA, we were using antibody coated wells where already some antibody, some specific antibody will be coated. Okay, so these wells here will add the patient sample. If the patient sample is having the antigen specific to this antibody, then it will bind with the antibody, and then we can either perform direct ELISA and we can perform indirect ELISA also. So if in case of direct ELISA, what will happen? If the patient sample is having the specific antigen, then the antibody, enzyme conjugated antibody will bind, and then chromogenic substrate will show the color, just like we have studied the direct ELISA. In case of indirect ELISA also, here we are using two sets of antibodies. The second antibody, the secondary antibody, will be bound with the enzyme, and then the chromogenic substrate will show us the color. So ultimately here, by observing the color, we can know that antigen was present in the sample or not. Here it is called sandwich because the antigen, if you see, it is stacked in between two antibodies. Here it's just like the sandwich. In the sandwich, stuffings is there in between two breads. Similarly, the antigen is there in between two antibodies. That's why it is called sandwich ELISA. Last one is your competitive ELISA. What happens in competitive ELISA? Here, the conclusion is that if the signal or if the chromogenic substrate is showing us less color, that means the infection is more in the blood sample or in the body. If the signal is more, then the infection is less. If the signal is less, then the infection is more. Let's take an example that we had total 8 antibodies. In one test tube, there were 5 antigens. Suppose this is patient 1, this is patient 1 or sample 1, this is sample 2. In patient 1, he had 5 antigens, for example, 5 antigens, and the patient 2 was having 2 antigens. So patient 1 is having more infection, patient 2 is having less infection because less number of antigens were there. So with here, what we'll do, we'll add antibodies. First we will add the patient sample, then we will add some antibodies specific to that antigen which we were suspecting that the patient might be having. Then these 5 antibodies will bind with the 5 antigen. Total we had added 8 antibodies, but only 5 antibodies will get bound to the antigens, because here the patient is having only 5 antigens. So out of the total 8 antibodies, only 5 antibodies were bound to the antigens, because the patient was having 5 antigens. Now how many free antibodies we will have? We will have total 3 free antibodies. Bound or free antibodies we will have, because 8 minus 5 will be 3. Now what we'll do, we'll pour all this pre-mixture or pre-culture into another well. In this well, already antibodies will be coated, like we have already discussed about already coated wells, like antibody coated wells were there, antigen coated wells are there. Similarly, here also we are using antigen coated wells in wells where previously antigens will be coated. Now we'll pour all this pre-mixture into these wells. Now these wells are having the specific antigens. Now here 3 unbound antibodies were there, so only 3 antibodies will be able to bind with the antigens, and will use secondary antibodies conjugated with enzymes. Then we'll add the substrate, chromogenic substrate, color will be detected, and here we'll see less color or less signal. Why? Because less number of antibodies were bound, so less number of chromogenic substrate will give us color. These steps are similar with the indirect ELISA. Now in this case, what is happening here, infection was less, that means less number of antigens were there in the patient's blood. 2 antigen were there, total 8 antibodies we have, so only 2 antibodies will bind with the antigens, so 8 minus 2 will be 6. 6 left or 6 unbound or 6 free antibodies will be there. Now let's pour all this pre-mixture into the antigen coated wells. Now what will happen, total 6 antibodies will be able to bind with antigens. So 6 will be definitely more than 3, right? So 6 secondary antibodies will also bind, 6 enzymes will also bind, and 6 substrate will also bind, and thus more chromogenic substrate will give us color. So here we'll see more color. Now more signal is indicating less number of antigens or less infection. Less signal is indicating more number of antigen or more signal. I hope this is clear. If there is any kind of confusion, you can comment down below, or if you want to take detailed coaching from me for CSIR NET, SLATE or any life science exams, you can comment down below, or you can contact in the given number. I hope the video was helpful enough. So please subscribe to my channel if you like my videos, and share the video with your friends or anyone, any aspirant that you know. If you like my videos, then please subscribe to my channel.